Now we will discuss about the shoot and root apical meristem. So before going into detail, I'll give you the idea about what is meristem. So meristems are the cluster of cells which give rise to the body plan, okay, which forms the body plan of the plant, and they are similar to the stem cells in animals. Okay, these stem cells are the uncommitted cells or undetermined cells. We know that. Okay. Now, these meristem cells fall into three categories: apical, lateral, and intercalary. Apical meristem is located towards the apex side. It helps in the plants to become taller. Intercalary meristems are located towards the medial region, so the mid region, and lateral meristem is located towards the sides. Okay, this increases the girth or the width of the plant. Okay, now here I have included a full picture for you to understand it more clearly. See this, the upper part is shoot apical meristem, which I'll write as Sam, a nice name. Okay, so this is Ram. Okay, another nice name. So Sam have the terminal bud region. Okay, the Sam will make the plant taller. Okay, and Ram will help the plant to grow its root. Okay, so Sam and Ram both have a good connection. Sam helps the plant to grow from above the soil, and Ram helps the plant to grow below the soil. Clear? So we will first elaborately discuss about shoot apical meristem. So shoot apical meristem is present here, and this is the dome shaped structure. Okay, the shoot apical meristems, they are usually indeterminate in their development. That means they are not determined. Okay, they are indeterminate in their development, and they can be converted into floral meristem. Okay, when the plant is induced to flower. So during induction, or during when the plant is induced, then these shoot apical meristem can form the floral meristem. Okay, that means they are not determined. They can form floral meristem. So once floral meristem is formed in this region, which is in the dome shaped region, then this meristem is the determined one. They cannot form the vegetative meristem back, which is the shoot apical meristem back. Okay. Just a moment. We will discuss about the floral meristem in detail. Now, so floral meristem are determinate. So see, so first I will give you the overview about the development of shoot apical meristem, then we will go in detail. So see, I have clipped this picture from a book, and I'll give you the little idea about what happens in the shoot apical meristem, how they are formed. Okay, so see, this, there are three zones, which is the organizing center, central zone, and the peripheral zone. I have highlighted to catch your eyes in these regions only. Okay, so see, there are three regions: organizing, central, and peripheral zone. Okay, the here what we see, so this is the organizing region, peripheral region, and the central zone. We see that WUS genes, which is the full name is WUSCHEL gene, we'll discuss in detail, do not get over it. The WUS gene, the short form is WUS. The WUS genes and the clavata genes, CLA, which we will write as CLV. Okay, so the WUS genes and clavata 3 genes. Clavata genes are clavata 1, clavata 2, clavata 3. So WUS genes and clavata 3 genes, they helps in the organization of shoot meristem. Okay, now WUS gene is expressed where? WUS gene is expressed in the organizing region. In the organizing center they are expressed. Okay, and they induce, or they help the stem cells, or they help the cells to remain in the undifferentiated state. I have written here, you can go through the slides later, that WUS genes help the cells to remain in undifferentiated stem cells. Okay, they do not help the cells to become determined. Clear? Now this WUS gene, they activate CLV gene. Okay, WUS gene then activate CLV3 gene. This CLV3 gene, when activated, it produces CLV3 protein. This CLV protein serves as a ligand. Where are the receptors for this CLV ligand located? They are located in the peripheral region. Okay, we will discuss in detail. I am just giving you the overview right now so that you can relate. It will not confuse you further. So CLV gene will bind to the receptor in the peripheral zone. So once CLV3 gene binds to the receptor in the peripheral zone, then it limits the expression of WUS in this region. Okay, see the inhibiting arrow. Okay, so once CLV3 gene binds to its receptors in the peripheral region, they do not allow the WUS expression in the peripheral region. So it is limiting the WUS expression in the peripheral region. So as a result of which, what will happen? That a small population of stem cells is limited to the growing tip only. Okay, there will be no stem cell in the peripheral zone. The small population of stem cell will be limited to the shoot tip region only. Clear? And the peripheral zone are allowed to grow and differentiate. The peripheral zone are allowed to grow and differentiate into lateral structures, which is leaves, but not the stem cells. Clear? Now, let's discuss in detail. Now, two different concepts have been used to define the regions of the shoot apical meristem. So, one concept is of layers. So, one concept is of layers. So, first concept is of layers. Two different concepts have been used to describe the shoot apical meristem. So, first concept we will discuss about is of layers. So, according to this concept, it is seen that the shoot apical meristem has three layers, L1, L2 and L3. The L1 is the outermost layer, okay, and cell division in this layer will form what? It will form epidermis, okay, and the L2 and L3 layers, they both forms the internal organs, okay. So this is the first concept of the development of shoot apical meristem. So it was found that while cells in certain layers tend to have certain fates, okay, certain layers have certain fates, but they are not committed. Okay, if a cell is shifted to a new layer, it generally develops like the other cells in that layer. That says that the shoot apical meristem cells are not committed. Next, see, so if this layers are not developed properly, then it give rise to chimera or variegated leaf. So see, so in this plant, which is a tobacco plant, it was seen that the L2 layer here, it lacks chlorophyll. Okay, L2 layer lacks chlorophyll, which gave rise to the chimeric plant, and this is known as, this has the kind of leaf it is showing is known as variegated leaf. Okay, in the wild type, it is seen that the L1 layer lacks chlorophyll except the guard cells. Okay, just to have the idea, the question does not come from here, just to have you the idea. Okay, but in this picture, the L2 layer also lack chlorophyll. Okay, and L3 remains green. Here the L3 does not contribute to the outer edges of the leaf. That is why the outer edges appear green. Since L3 is green in color in the wild type, but here L3 is not there in the outer edges. So that's why the outer edges are also white in color. Clear? Now see, this is the first concept, which is layers. And the second concept is of histological zones. Clear? So we will study about this here. So see, longitudinal section through the center of the shoot apex. Okay, what it shows that shoot apical meristem have three cell layers, which is L1, L2 and L3. I write here L1, L2 and L3. Okay, L1, L2 and L3. Okay, Now, this L1 will form epidermis and L2 will form shoot epidermis, and L2 and L3 will form internal tissues. The shoot apical meristem also has cytohistological zones, which are peripheral, central, and rib zone. Okay, we see that the peripheral zone in which the cells will divide rapidly. Okay, in the peripheral zone, there will be the rapid cell division. This peripheral zone is surrounding the central zone, and this peripheral zone will produce what? It will produce leaf primordia. That means from where the leaves will be developed. Okay, the initial cells from where the leaves will develop. Okay, another one, the rib zone. The rib zone is what? It is located below the central zone, and it generates the central tissues of the xylem. Okay, sorry, central tissues of the stem. Now coming to the central zone, its central zone contains what? This is a very important zone. They contain stem cells. Central zone contains stem cells. These stem cells divide very slowly. Okay, slow division. And they are the ultimate source of the tissues that make up the plant body. So this is the concept of zones, and this is a concept of layers. Clear? Now we need to know the genes which control the shoot apical meristem. I have not written the full form. So the genes which control the shoot apical meristem are STM gene, which is shoot meristem less gene, Wuschel gene, and clavata. We discussed the genes which control embryogenesis about shoot meristem less, that they helps the shoot to remain in the undifferentiated stage, to remain in the undetermined or uncommitted stage. Okay, Wuschel also do the same thing, and clavata. The clavata genes play an important role in limiting the number of undifferentiated cells. This clavata genes limit the number of undifferentiated cells. And this STM and Wuschel then genes, what they do, they keep the cells in the undifferentiated state. So kind of we can say that the clavata is having the opposite role than the STM and Wuschel. Okay. So see. So in the normal condition, what we see that STM and Wus means Wuschel, STM means shoot meristem less. Okay, they will keep the shoot in the undifferentiated state, and clavata genes will limit the number of undifferentiated stem cells or the meristem cells. Okay, now STM prevents the differentiation of stem cells independent of Wus, means Wus is independent of STM and STM is independent of Wus. They both keep the meristem in the undifferentiated state. And Wus expression defines the organizing center. We discussed that they are present in the organizing center. Okay, and they promotes the cell division. Okay, they promotes the cell division and they defines the organizing center. Now when there is the mutation in these genes, so when Wus and STM are mutated, there will be no seedling. Okay, with no or reduced SAM. Okay, I'm sorry. So when there is no STM or Wus, then it is seen that the SAM or shoot apical meristem is either not formed, or when it is formed, it is reduced. Clear? So mutation in STM, it results in embryos without a SAM. We already discussed in the first point. Okay, but when CLV is mutated, it results in enlarged SAM. Why? Because CLV's role was to keep the cells in the or limit the number of undifferentiated cells. So if CLV gene is not there, then that there is no limitation in the formation of the undifferentiated cells. So undifferentiated cells will be produced in large number. Okay, so which give rise to enlarged SAM. Okay. Now, so I would like to let you know one more thing, that where are these STM genes expressed? Okay, so STM genes are the shoot meristem less genes. They are expressed in the shoot apical meristem region. Okay, but whose gene is expressed in the organizing region also I discussed with you. See, whose gene is expressed in the organizing region also. But STM gene is expressed only in the shoot meristem region. Okay, STM gene is expressed only here and not anywhere. It is not expressed in the peripheral zone. It is expressed in the shoot apical meristem zone. Next, we will discuss in detail about the clavata gene, Wuschel gene, and STM gene. Okay, so when this clavata gene, okay, so mutation in the Arabidopsis clavata genes led to increased meristem size, because clavata genes limit the number of undifferentiated cells. So when these clavata genes are mutated, then meristem size will be enlarged and there will be the production of extra organs. Okay, so there are three clavata genes, 1, 2 and 3. They limit, all of three limits the number of undifferentiated cells. Okay, so that there should be limited number of cells to be produced. Okay, now this clavata1 gene, okay, they encodes a receptor. Okay, these receptors are kinases, serine threonine kinase. So clavata1 gene encodes a receptor, and its ligand is what? The ligand of clavata1 gene is the product of CLV3 gene. So CLV3 gene will produce a protein known as CLV3 protein. This CLV3 protein acts as a ligand. And who is the receptor of this ligand? The receptors are CLV1 and CLV2, which are the receptors. Okay, they both are the receptors for this ligand. Clear? Now, the peculiarity of CLV1, clavata1, is that they have the receptor with the kinase domain. Okay, now clavata2 is also a kind of receptor, but they lack kinase domain. Which kinase? Serine threonine kinase. Okay, clavata1 has serine threonine kinase. Okay, so once clavata1 binds to the CLV3 ligand, then there is a signaling pathway which is induced. The signaling pathway, when it is induced, it will limit the number of the undifferentiated cells. Okay. So a CLV3 restricts its own domain of expression to the central zone by preventing the differentiation of surrounding cells. We will see this later. So STM, Wus and Paul genes. So there are another genes named as Poltergeist and Wuschel genes, which keeps the cell in the undifferentiated state. STM, Wus and Paul, all three keeps the cell in the undifferentiated state. Okay, so Wus specifies the stem cell identity in the cells positioned above. Okay, and when the CLV signaling pathway starts, it shows a negative feedback loop on Wus expression. Once CLV receptors are activated, then they inhibit the Wus expression. Okay, once Wus expression is inhibited, the size of the meristem is regulated. Clear? So STM, Paul and Wus, they all three play important role to keep the cell in the undifferentiated to keep the tissue in undifferentiated state. Okay. Now, so the interaction between clavata genes and the STM, Wus and Paul genes, what do? That they balance the rate of cell division. Clear? And the rate of cell differentiation. So look here, the presence of Wus gene. So which region is this? This region is the organizing region. So in the organizing center where the Wus genes will be produced, this Wus gene will activate the clavata3 gene. See the green arrow which is going. I change the color here. See, the Wus gene is activating CLV3 gene. The CLV3 gene is producing what? CLV3 protein. The CLV3 protein is a ligand, okay, which binds to their receptor. Where are the receptors of the CLV3 located, they are located in the L3 layer. Okay, in the L3 layer, it will go and it will bind to its receptor, which is either clavata 1 or clavata 2. Once they are binding to its receptor, they are inhibiting Wus expression. Okay, they are inhibiting the Wus expression in which region? In the peripheral region. This is the peripheral region. So clavata 1, 2 is inhibiting the Wus expression in the peripheral region. When are these clavata proteins, clavata 1, 2 receptors are activated? They are activated when they bind with clavata 3. Who activates clavata 3? Wus activates clavata 3. So Wus is such a good person that it is helping somebody, and this person, okay, is backfiring on Wus. Clear? So this creates a negative feedback signal, okay, which will inhibit the Wus expression, so that the cells cannot be in the undifferentiated state any longer. Okay. So what we studied, that Wus will activate. So arrow will indicate activation. Okay. Wus will activate the clavata 3 gene. This clavata 3 gene is where it is expressed in the L3 layer. See here. This clavata gene here. So here I have included L3 layer. So actually it will not be L3 layer. Why I have written, I don't know. So it is this CLV3 gene. Okay, this is expressed in L1 and L2 layer. Okay, they are expressed in L1 and L2 layer. Okay, so once this CLV3 gene is binding to its receptor, we see that they inhibit the Wus expression. And where are these Wus genes expressed? These Wus genes are expressed in the small number of cells in the L3 layer only. Okay, in the L3 layer, Wus genes are expressed. Okay, so what we need to know till now, what we have studied till now, we have studied that Wus genes, STM genes, which is shoot meristem-less genes, and Paul gene, they three help in help the cells to remain in the undifferentiated state. Okay, Wus is expressed in the organizing center. They activate the CL3 gene. The CL3 gene will produce CLV3 protein. The CLV3 protein acts as a ligand for CLV1 and CLV2. So CLV1 and CLV2 acts as a receptor. Once the receptor becomes activated, then they inhibit the Wus expression. Okay, so they inhibit the Wus expression in the peripheral zone, so that Wus expression is limited only in the shoot apical meristem zone. Clear? This is we need to know in the whole pathway. So here I have included, see that the Wus is activating CLV3, and CLV3 when it binds to CLV2 or CLV1, they inhibit the Wus expression. Clear? Now a small chart I have created here. See what we see that CLV1 and CLV2 3, when all are mutated, because they limit the undifferentiated state, so when they are mutated, the resulting structure will be the bigger SAM, because more and more cells will be produced, more and more undifferentiated cells will be produced, so it will be enlarged SAM. When CLV3 is high, okay, when CLV3 is high, we see that the function is normal. Okay, when when CLV3 is high, then there will be the normal function. Clear? Now, when Wus is high, or when Wus is mutated, that means the cell cannot be in undifferentiated state. This will be smaller SAM. Okay, when CLV1 and Wus both are mutated, okay, then there will be also smaller SAM, because once Wus is mutated, then there is no need of clavata 1. Clavata 1 will inhibit with G. Okay, so when Wus is inhibited, look at Wus only, there will be smaller SAM. Again in the second stage, when Wus is inhibited, smaller SAM. When Wus is inhibited, smaller SAM. But whenever the CLV gene is high, okay, whenever the CLV3 is high but CLV1 is mutated, So since CLV1 is mutated, then we see that there is no limitation to the growth of the SAM, so it will be bigger SAM. Now when WUS is very very high, the formation of undifferentiated cell will also be more over proliferation. When CLV1 and CLV2 both are mutated, okay, when CLV1 and 2 are mutated, CLV3 is produced too much, so there will be bigger SAM. Clear? With that, we will solve one question. Okay. So see, here in this question, it is saying that loss of clavata1 gene leads to bigger SAM. Okay. So again, so based on this, we need to find out which of the answer is correct. So some options are given to us. So loss of clavata gene leads to bigger SAM. So when clavata1 is lost, then what will happen? There will be no limitation because they limit the number of undifferentiated cells. So no limitation to growth. So there will be bigger SAM, yes. Clavata3 genes. So loss of clavata3 genes. When it is lost also, then what we see, it leads to bigger SAM. So here we do not have to find which option is correct. We have to see that among these options, which fits best to our answer. Okay, so loss of Wuschel gene leads to smaller SAM. So Wuschel helps in undifferentiated stage. So if Wuschel is not there, then it is smaller SAM, yes. Okay, so see here. We among this, okay, let's me read first. The loss of both CLV1 and WUS leads to smaller SAM. Loss of CLV3 and WUS leads to smaller SAM. Okay, so you try to make a pathway on your own. So loss of both CLV1 and CLV3 leads to bigger SAM. Overexpression of CLV3 leads to smaller SAM. Overexpression of CLV3 in the loss of function mutant CLV1 leads to bigger SAM. Okay, based on the above information, which of the following genetic pathways describe the relationship among CLV1, CLV3 and WUS? Okay, so what we know, if I do not look at the answer, what I know, that WUS will activate CLV3. Okay, this CLV3 will bind to what? It will bind to either CLV1 or it will bind to either CLV2. Okay, once they bind to this, they inhibit WUS. Clear? So which pathway seems correct? So here it's saying that clavata1, 3, they are activating WUS. This is not. Clavata1, 3, it is inactivating WUS. And WUS is regulating SAM size. But clavata3 is not inhibiting WUS. This is wrong. Third, it's saying that clavata3 will activate clavata1. It can activate clavata1 also. It can activate clavata2 also. Okay, clavata1 will inhibit WUS and WUS regulates SAM size. So this seems correct, right? Fourth one says that clavata1 inhibits clavata3. No, this is totally incorrect because clavata3 activates clavata1. Okay, clear? So third is the right answer. Let's discuss about the root apical meristem. So root apical meristems are present. So root apical meristems, they are the population of cells at the root tip. See, at the root tip, they are the population of the cells and they will form all the tissues of the root. Clear? Now these root apical meristems, they are the population of dividing cells. But not all cells in the meristem, they divide at the same rate. So which cells do not divide at the same rate? There are the cells known as the cells in the quiescent center. Okay, so look, the cells in the quiescent center, they divide very slowly. Okay, they are very slow dividing cells. The question comes from here. Quiescent center, the term you should be familiar with. Clear? Now, this quiescent center are the slow dividing cells, and Arabidopsis, the quiescent center consists of only 4 cells. I'll show you the picture. So remember this here, the meristematic zone, which is the zone which will divide further and keeps the cell in the undifferentiated stage. Okay, the elongation zone where the roots will elongate from, and the maturation zone. Clear? The maturation zone will form several thickenings. See, the root hair, you can see the root hair are coming from which zone? They are coming from maturation zone. The root hairs are coming from the maturation zone. Okay, which is beyond the root tip. Clear? Also, we see that at the tip of most of the root is what is the structure known as root cap. Okay, this root cap helps in protection of the underlying root meristem. Okay, they protects the underlying root meristem, which is just above it. Okay, so all the species with some exception produces the root cap. Clear? Now it was found that auxin plays an important role. Okay, auxin plays an important role in the formation and maintenance of ram. Okay, so auxin plays an important role in the, since auxin helps in the formation of root, so it plays an important role in the formation and maintenance of ram. Ram means root apical meristem. Clear? So this is all about the root apical meristem. Now look at this picture also. I told that in Arabidopsis there are only 4 cells in the quiescent center. That is why I have put this picture here. Okay, see, there are only 4 cells in the Arabidopsis quiescent center. Okay, and there is slow growth in the quiescent center. This is I want you to show. Clear? So there are different layers: outer epidermis, cortex. Please remember the sequence. I already discussed this when I was doing the transportation. Okay, epidermis, cortex, inside is the vascular bundle. Okay, surrounding the vascular bundle is pericycle, endodermis. Okay, so epidermis, cortex, then it's the brown colored one is the endodermis. Epidermis, cortex, then endodermis, then pericycle. Inside to the pericycle is the vasculature. Okay, so immediately following the vasculature is the pericycle. Clear? Now we will continue. We will go to another topic, transition to flowering. Okay, now this transition to flowering, like from the vegetative state, how the meristem becomes determined, how they form floral meristem. So from the juvenile stage, how the plants enter into the reproductive stage. Okay, juvenile means they are still young, they are not reproductive. That means they are vegetative. So from the juvenile stage, how they enters the reproductive stage, that we have to study. Okay, this is the transition to flowering, and flower is the reproductive organ of the plant. So if we discuss about the juvenile stage, where the plant remains in the vegetative stage, in the juvenile stage the plant cannot reproduce. Okay, they cannot form flower. So to convert or to transform from the juvenile stage to the reproductive stage, the plant needs to be competent. Competent means they must be able to respond to the signal. Okay, the signal can be internal and the signal can be external also. It depends on the plant only. Okay, so signal can be internal or the signal can be external, and the plant must be competent. See, this word is very important. Just a moment. The plant must be competent to the signal to become or to enter into the reproductive stage. Now in maize it was found that there is a gene which is responsible for the maintenance of the juvenile stage. This is known as early phase gene. So as long as this early phase genes are expressed, the plant will remain in the juvenile stage. Okay, so early phase genes, which is in short EPC, this maintains the plant in the juvenile stage. So if this EPC, which is early phase gene, if it is mutated, then the plant shows the change of phase very early. Okay, they flower very early because EPC maintains the plant in the juvenile stage. When this EPC is mutated, the plant flowers early. Okay, another important gene known as hasty gene. Now this hasty gene, what it does, it has also the same role like EPC. They also maintains the juvenility. Okay, so when hasty gene is mutated, we see that there is early phase change or early flowering. Okay, so hasty genes are released from the nucleus. Okay, so if this is a cell and this is nucleus, so hasty genes are required to release from the nucleus. Okay, to show it, to show its expression. Okay, so they are required to release from the nucleus to show its expression here. But when they are mutated, then HST gene cannot come out of the nucleus. They cannot come out of the nucleus. Okay, they remain trapped in the nucleus. So when they are trapped in the nucleus, they cannot maintain the juvenile stage. They cannot come out of the nucleus. So this is a cell, this is a nucleus. So to show its function, they need to come out of the nucleus. So they cannot come out when they are in the mutated stage. When they are in the mutated stage, there is early flowering. Clear? So look in this picture. Here it's seen that HST and EPC are expressed in the apical meristem and keep the cells in the vegetative state. Clear? And rest of the genes we will discuss in some time. So let me tell you in brief that there is one gene named as terminal flower 1 gene. Okay, TFL stands for terminal flower 1 gene. Terminal flower locus 1 gene. Now what this gene do? This inhibits the flowering. Okay, they inhibit the flowering. Another one, FT plus FD. When flowering, okay, I'll tell you the full form later. The FT and FD, when in combination, they activate the gene leafy and apetala 1. What they are, we will discuss in detail. I'm giving you the overview that FT and FD, they are when bind together, they activate LFE, LFY and apetala 1. LFY stands for leafy. All should be written in capital. Okay, and AP1 is apetala 1. This is just the overview. The apetala 1, when they are activated, the plant shows flowering. So leafy and apetala 1 are the genes which converts the shoot apical meristem into the flowering meristem. Clear? So see, they are the floral meristem identity genes. Leafy and apetala 1, they are the floral meristem identity gene. The role of floral meristem identity gene is to form flower. From shoot to flower. Okay. Okay, this is the role of floral meristem identity gene. Now, floral organ identity gene we will discuss in detail. So CO constants, when the light is there, the constants gene is there, CO, which stands for constants. This constants gene is activated. This constant gene activates what? FT. Okay, FT when binds with FD, they activate the, or they can, you can say that FT and FD helps in flowering. Clear? So we can see that 4 different pathways are involved here in the flower formation. So see, number 1. So phase change gene should be there, which helps in the juvenile vegetative growth to adult vegetative growth. Adult vegetative growth, remember this stage. Adult vegetative growth will form inflorescence, then there will be the formation of flower. First inflorescence, then flower. So in the inflorescence growth, there are numerous small leaves appear. These small leaves will form what? Flowers. Okay, so photoperiodic pathway, autonomous pathway, vernalization pathway and gibberellin pathway, all of the 4 pathways activate the gene named floral meristem identity gene. What is the role of floral meristem identity gene? They convert the shoot or the adult shoot, adult vegetative plant into the flower through the inflorescence growth, then the flower formation. So remember, the floral meristem identity genes have the role to convert the shoot to flower. Clear? And floral and inflorescence growth will form the flower. Okay, we will discuss about it in detail. For now, you remember that these 4 pathways are involved in the flower formation. Next, what we see, we saw that terminal flower formation, which is when suppressed. Okay, So the inflorescence meristem, it arises through the action of the gene that suppresses terminal flower formation. Not to be bothered about, we will discuss about it in detail. Next, what we see that TFL1, which is terminal flower locus 1 in Arabidopsis, and its homologue. Okay, I changed color. And its homologue. So centroradialis is a homologue of TFL1. Okay. What do? They inhibit the floricola gene. This floricola gene is what? It is also seen, this floricola gene is also seen in Arabidopsis. Okay, I'll discuss about floricola gene. So this centroradialis gene and TFL gene, they inhibit the flower formation. So how they will inhibit the flower formation? They will inhibit the floricola. Okay, floricola helps in flowering. So they inhibit the floricola. Floricola helps in the flower formation, terminal flower formation. So centroradialis and TFL, they inhibit the terminal flower formation by inhibiting the floricola gene. Okay, so CEN1 and TFL1, they delays the commitment to inflorescence development. Okay, they will delay the commitment to the inflorescence development. So if this TFL1 gene is overexpressed, okay, so TFL1 gene helps in inhibition of flowering. So when they are overexpressed, then the time to flower will be extended. Clear? Now the role of TFL1, I am again revising, its role is to helps or its role is to inhibit flowering. So it delays the transition to reproductive stage. Clear? Next we come to floral meristem and floral development in Arabidopsis and Antirrhinum. Antirrhinum is what? Antirrhinum is snapdragon. Okay. Lastly, we discussed about the transition from the shoot apical meristem to the floral meristem. Now we have to discuss about the floral meristem and floral development. Okay. Now see in this picture the four holes of a flower. We discussed earlier that flower have four parts which are known as holes. The four holes, sepal, petal, stamen and carpel, they are arranged in concentric rings, if you can see. They are arranged in concentric rings. So in wild type Arabidopsis, Arabidopsis flower, the holes are arranged as, see, the first or the outermost hole is the sepal. Okay, the outermost, I am showing about the concentric ring only. The outermost hole is the sepal, which is the green in color. Okay, next the second hole is petal, which is colorful and it is white at maturity, as we are talking about Arabidopsis. The third hole is the stamen. So here it is, Arabidopsis has how many stamens? 6 stamens. See, the blue colored ones are stamens, 1, 2, 3, 4, 5, 6. Arabidopsis have 6 stamens, of which 2 are shorter and 2 are longer. We do not have to learn that. The fourth hole is the gynoecium or the pistil, which is composed of ovary, style and stigma, which is towards the interior side. Okay, carpel. So this model, which is known as combinatorial model, it shows that each hole are determined by overlapping developmental fields. Okay, see the field are overlapping, field 1 and 2. So sepal and petal by field 1, petal and stamen by field 2, and stamen and carpel by field 3. We will discuss about it in more detail. Do not get confused. Okay. Now another very important part. So in any case you are not going to miss that for your exam. Okay. In any case you will not miss it, because every time, each and every time, be it part B or part C, CSIR net, this question comes. So this is a favorite topic of all the entrance exam. If this is in your syllabus, this is a favorite topic of the examiner. Okay. So let's study. So see, 3 types of genes are there which will regulate the floral development. What they are? Floral organ identity gene, cadastral gene, and the third one is meristem identity gene. What is the role of floral organ identity gene? They control the floral identity. Okay. They produce the products that are involved in the formation of floral organs. What they are, I'll discuss. I'll tell you. I've made a beautiful chart for you so that you will remember at once only. Now, cadastral genes, they are regulators of the floral organ identity genes. So here in your life, like I'm telling you about the similarity. So cadastral gene will set the boundary. It will say that you have to express till here only. You do not have to go further. At sepal, you have a limitation. You cannot take the petals area also to form sepals. Okay, petals have the limitation. They cannot take the stamens area. Same way, stamens cannot take the area where the carpels can form. So who will limit? There must be some genes which will limit the formation of the organs. So these are cadastral genes. So in your life also we play the role of cadastral genes. We tell you that you have to study this much only. We will define the areas that do not go beyond that. Okay, some students still have the question that like they study far beyond the what is the syllabus is, and they take lot of time to complete the syllabus. Okay, they one of the topic they like so much that they enter into the depth of that topic and they forget about that they have to complete the other topics as well. Okay, so the teachers play the role of cadastral gene. They define the boundary that stay in this boundary, you have to study this much only to qualify the exam. Clear? So one example, it was found that Superman gene. So teachers play the role of the Superman. Okay, very funny. Now the Superman gene, they are the example of cadastral gene which specify the boundary for the expression of organ. Okay, like they fastens your process, right? Now, meristem identity genes, they are the ones which helps in initial induction of organ identity genes. Okay, so meristem identity gene which will help in the formation of organ identity genes. Okay, so these meristem identity genes helps in the formation of floral organ identity genes. They activate, or they are the positive regulator of floral organ identity genes. Okay, and floral organ identity genes will be limited by their expression, will be limited by, who will limit their expression? It will be limited by cadastral genes. Clear? See, I've made such a beautiful chart, a very clear and well defined explanation so that it will help you to remember the names of the genes all at once. Okay, so three types of genes we studied: floral meristem identity or floral meristem identity. Both are same. Okay, floral meristem identity. Okay, both are same. So floral meristem identity genes will activate what? They will activate the floral organ identity genes. Okay, they are the positive regulator of floral organ identity genes. So floral meristem identity genes, what is their role? That they help in the conversion or the transition from shoot meristem to the floral meristem. Okay, so from shoot to flower, floral meristem, the genes involved are leafy, LFY, apetala 1 and cauliflower. These three help in the formation of floral meristem from shoot meristem. Please remember this. I'm giving the star mark, double star mark. I'm writing important by its side. In any case, you please do not forget. This is very simple concept. Now, floral organ identity gene, they control the floral organ identity, like which gene will form which organ. Okay, which organ is to be formed in which region is controlled by floral organ identity gene. So these are 5 in number: apetala 1, 2, 3, pistillata, agamus 20. Sorry, agamus only, agamus. So they define the organs, which organ is going to form in which region. Clear? Floral meristem identity gene will help in the transition from shoot meristem to floral meristem, which are leafy, apetala 1 and cauliflower. Okay, you can see that apetala 1 is here. I've added it here also. So apetala 1 is floral organ identity gene also. Please remember, I'm giving the star mark. Apetala 1 is floral meristem also. Apetala 1 is floral organ also. Okay, next see. So see, there are two classes of floral meristem identity gene. One will help in the floral meristem formation. What they are? The upper ones which we discussed in last slide: leafy, apetala 1 and cauliflower. Cal stands for cauliflower. Okay, and the opposite effect, which is the terminal flower, which will inhibit flowering, we discussed in the last few slides. Terminal flower 1, TFL, terminal flower locus 1, it has the opposite effect, it inhibits flowering. Clear? Its homologue is what? Its homologue in Snapdragon. Snapdragon scientific name is Antirrhinum, which is given in the syllabus. Do not get confused. Homologue is Centroradiolus in Snapdragon. Next. So see, there are three classes of genes in floral organ identity genes. Three classes. Class A will express, I said there are overlapping fields. In the first slide when I started, I've shown you the diagram where there are concentric circles and the fields are overlapped. So in the first field, class A gene will be expressed. Class A gene include Apetala 1 and Apetala 2. Class B gene include Apetala 3 and Pistillata. And class C will express only Agamus. So do not worry, we'll solve several questions based on this, so that you will learn in my class only. You do not have to give extra focus. Now this also we discussed, that meristem identity genes, what it do, it converts the, or it helps in the transition of shoot meristem to the floral meristem. Okay, antirrhinum, which is snapdragon. Okay, mutants of antirrhinum that have defect in the meristem identity gene floricola develop an inflorescence that does not produce flower. I told that FLO gene helps in flowering. So when FLO gene is what? FLO gene is seen in antirrhinum, snapdragon. It helps in flowering. So when this gene is mutated, there will be no flowering. Okay. So mutant floricola gene results in the development of additional inflorescence meristem at the bract. Okay, now the wild type floricola gene, it should be in capital. I'm very sorry, it should be in capital. All the genes will be in capital. Now the wild type floricola gene, it controls the determination step. Okay, and it helps in the formation of floral meristem. Clear? Now, leafy is the homologue. Sorry. Now, leafy gene is the homologue of this FLO gene in the floricola. Sorry, in the Arabidopsis. So, leafy is the homologue of FLO gene. Okay. And TFL1 in Arabidopsis is the homologue of Centroradiola gene we discussed. Do not forget, please. Now, Agamus L20, very important. This gene is very important. Okay. Agamus L20, it plays a central role in floral evocation. It integrates the signals from both environment and internal cues. It's a very important gene which combines the signals, or which synchronize the signal from both environmental and internal cues. They act as a master switch in floral development. So once this AGL20 is activated, it triggers the expression of LFY, and LFY turns the expression of AP1. Okay, and this LFY and AP1, what are their roles? The role of LFY and AP1, that they helps in the floral meristem formation. Okay, or floral meristem identity. They are floral meristem genes. So in Arabidopsis, LFY and Apetala 1 are involved in positive feedback loop. That AP1 expression also stimulates the expression of LFY. So LFY also expresses AP1, AP1 also expresses LFY. So both acts as a positive, or both forms a positive feedback loop. So AGL20 is a master switch, remember. Now, another important part is the homeotic gene. So when there is homeotic mutation, it led the study of the homeotic mutation led to the identification of floral organ identity genes. Okay, so see here, mutations in Drosophila led to the identification of set of homeotic genes. Okay, I changed the color because red is mixed here. See, these homeotic genes will encode the transcription factor which will determine the location at which specific structures develop. So in a simple language, I can say that when there is a mutation in the homeotic gene, it determines the location of specific structures, or the location, the specific structures where these specific structures develop. Okay, so this homeotic genes gives organs their identity. Okay, now the homeotic genes in plants, they belong to mad box genes. Okay, where in animal the homeotic genes are, they contain sequences called homeoboxes. So there is a, here lies a difference between animal and plants, that the homeotic genes of plants are related to the sequence of mad box genes, but in animals they are related to the sequence called homeoboxes. Okay, now we need to know about the discovery of mad box genes. So mad box genes is a conserved sequence motif. Okay, the genes which contain this motif are called mad box genes. Okay, so any gene which has a sequence similarity with the mad box genes, with the mad box, is are known as mad box gene family. They are included in mad box gene family. Okay, so how this mad gene have been discovered? So M came from MCM1, which was found in yeast. A came from agamus, which is found in Arabidopsis, and D stands for deficiens, which is found in snapdragon, and SRF is found in humans. So this is how the name MADS came. Okay, so any sequence which has a similarity with the mad box gene, they are included in the mad box gene family. Homeotic genes have the similarity with the mad box gene, so they are the class of mad box genes. Okay. Now, many of the genes that determine floral organ identity are mad box genes. Please remember this. Now, deficiency is the one. Agamus, I told you, is the one. Pistillata and AP3 genes of Arabidopsis, they are also mad box genes. Okay. Now, not all genes containing the mad box domain are homeotic genes. So, these are the homeotic ones. These are the homeotic ones. But all the genes which have mad box, they are not the homeotic ones. Okay, so AGL20, this has mad box genes, or this has a similarity, or they are included in the mad box family gene. Okay, they are included, but they are not homeotic genes. They are not homeotic. Okay, now look here. So 5 different types of genes are identified which controls the floral organ identity. What they are? They are Apetala 1 and Apetala 2. This Apetala 1 and 2, they are grouped under class A genes. I told you earlier. Apetala 3 and Pistillata are grouped under class B, and only Agamus is there in class C. What is the fate of these? I'll tell you. So when there is any mutation in these genes, the floral organ identity is changed. Okay, but the flower will form, like it happens in ultra bithorax and antennapedia gene of Drosophila. They are also the homeotic genes. The organ will be developed, but the identity will be changed. Okay, now these homeotic genes, which is I have mentioned here in above, they fall into 3 categories: type A, B, and C. You can say class, you can say type. Okay, now type A, which is encoded by Apetala 1 and Apetala 2, they shows their expression in the first and second hole. Okay, type B, which is encoded by Apetala 3 in Pistillata, they shows their expression in the second and third. So I told that the fields are overlapped, so they are expressed in first and second. They are expressed in third and second and third. Okay, and type 3 is in third and fourth holes. Clear? Type 3 is encoded by a single gene known as AG. Okay, So loss of activity, I'll tell you, I'll show you the chart, then I'll explain you. I've just told you about type A, B and 3. So when all the genes are mutated, so when one of the gene is mutated, the fate is changed. But if all the gene are petala 1 are petala 2, so remember the mutated gene we will write in small letters. So when all the genes are mutated, then it produces floral meristem which has developed pseudo flowers. Okay, in pseudo flowers, which we see that the floral organs are replaced with green leaf-like structures. Okay, so pseudo flowers produce flowers which have green leaf-like structures, and they have the hold phyllotaxy. So in all the holds, there will be the formation of the green leaf-like structure. Now, look here. Now, in type A, what were the two genes expressed? In type A, the genes expressed was apetala 1 and apetala 2. Okay, type A alone specifies sepals. Type both A and B are required for the formation of petals. Okay, I'll show you the genes in the next slide, so I'm not writing here. So type A specifies sepal, type A and B petal, A and B petal, B and C stamen, and C alone carpel. Okay, A alone specifies sepal, A and B petal, B and C stamen, and C carpel. Okay, so next C. Now, this model further proposed that the activities of A and C mutually repress each other. So A says to C that wherever you will go, I will not be there. Okay, wherever you will go, I will not be there. C says that wherever you will be there, I will not be there. Okay, they are like their enemies. If A is there, C will not be there. If C is there, A will not be there. But if C is not at home, then A will go to C's home to occupy the space. But in the presence of C, A will not go. But when C is not there, then A will go and occupies its space. I'll give you the I'll show you the picture, then it will be more clear. So remember for now that A is antagonistic of C. A inhibits C, C inhibits A. Both are antagonistic to each other. But B is such a friendly man. Okay, so he's such a friendly man. He says that I will with I cannot stay alone. Either A you come with me or C you come with me. I cannot stay alone. Okay, so he wants either A with him or he wants either C with him. Okay, he will not be expressed alone. But A and C have the ability to express independently. Clear? C. Mutations in the floral organ identity genes dramatically alter the structure. This is a wild type where the floral formula is totally perfect. So this for this structure we need not to remember, not required. So what we need to remember, I'll show you in the next slide. See, so this is the ABC model, which is very important, and this picture is also very important. Here what we see that there are three types. So the genes are grouped into three types: type A, type B, and type C. Type A express two genes. I'll change the color into green. See, type A expresses two genes, apetala 1 and apetala 2. So I write here type A is expressing what? Apetala 1 and apetala 2. Okay. Type B, in the second hole, the expression of both type A and type B results in the formation of petals. Okay, in the second hole, that is B, we see what is expressed. It is expressed another gene which is apetala 2 and pistillata. Now, in the third, we see in the third hole, the expression of B. Wait, in the third hole, the expression of B and C causes the formation of stamens. So see here apetala 2 and apetala 3 will be there. Wait, students. Okay, so since this is overlapping, in type A we will see that apetala 1 and apetala 2 is formed. So I erase from here, you look the ones I have written downward. Okay, see in type A, two genes are expressed, apetala 1 and apetala 2. This apetala 1 and apetala 2. So how we will see? First we will look at this chart which I have drawn. Okay. Second you will look at the picture. Thirdly you will look at the explanation. So look at the picture first. In type A, apetala 1 and apetala 2 are expressed. Okay. So apetala 1 and apetala 2 are expressed in type A. And type A alone is responsible for the formation of sepals. In the second hole, what we see that second is both type A and B. So A plus B. Second is A plus B. So here. So this is A. This is A plus B. Okay. A plus B will help in the formation of petal. Okay. A plus B will help in the formation of petal. For the formation of petal, which genes are required? AP2, AP3 and pistillata. Clear? Now, stamen, which is B plus C. I told B always expresses an association. So B plus C, the genes responsible are apetala 3, pistillata and ergamus. Okay, so apetala 2 came from the first one. Apetala 2 which was here, it came from here. Okay, and from B, which genes are there in B? In B there are AP3 and PI. Clear? Now, in the formation of stamen, AP3, PI and ergamus are required, which is B plus C. In the C part, which is carpel, only ergamus is required. So this is the overlapping field. You must have understood. Okay, so see, activity A represses the activity C in holes 1 and 2, while C represses A in holes 3 and 4. I'll again define you this, the last one in the next picture. Look here. So first we discussed what happens in the wild type case. Now we are discussing what happens in the mutant stage. Okay. This is again very important because the question are asked from the mutants most of the time. Okay, and all of the time. So see, in the wild type what we see that A gene, only A is responsible for formation of sepal, and A and B for petal. So A is for sepal, A and B for petal. Look what I am drawing in the picture. And B and C is for stamen and C alone is for carpel. Okay. Loss of C function. Listen to me very carefully. Loss of C function. When the C is lost. I told if C is lost, A will occupy C's space. So see, if C is lost, A have extended its legs. A have occupied the C's position. Okay, so wherever the A is alone, this region A is alone. So wherever A is alone, A will form sepal. In the middle region, A is with B. Okay, so A is with B. See the B's position have not changed. Okay, so wherever A is with B, the petals will form because A plus B forms petal. I told you only A will form sepal. When it is A plus B, it will form petal. So whichever region A and B are together, it will form petal. And the third region, the last region, we see that only A is there. There is no B above it. There is no C. So this will also be sepal. Clear? Now, loss of A function. I told you if A is not there, C will extend its leg. Okay? C will occupy the whole area. Clear? Now, this wherever B and C are produced, wherever B and C are together, I told this will be stamen. And whenever C is alone, it will produce carpel. So see the region where, please remember this diagram, learn this diagram by heart. Okay, just practice once or twice and it will be very clear. You can solve all the questions based on this. So see, whenever C is alone, it will form carpel. Whenever C is alone, it is form carpel. When B and C are together, it will form what? It will form stamen. Clear? Now, if B is lost, there are only A and C. Okay, if B is not there above A, then there will be no petal, there will be sepal only. If there is no B above the C, then there is no stamen, there will be carpel only. So I hope this is clear to each of you. Now, another very important pathway which we have to study. So see, phytochrome, cryptochrome, photoperiod all will activate a flowering hormone named as florigen. Okay, this is a flowering hormone. The flowering hormone will activate a gene named as constans. This will activate a gene named as constans. This constans act as a positive regulator of agamas like 20. I told this is a master switch. Once agamas switch 20 is activated, it will activate the meristem identity gene, another meristem identity gene which is leafy. This leafy will activate a petala 1. I told that AP1 and leafy can activate each other. Okay, they form a positive loop where they can activate each other. I told you that AP1 is meristem identity gene also, it is organ identity gene also. Okay, they will activate each other. Okay, so leafy once activated, it will activate all other organ identity gene or floral homeotic genes or floral organ identity genes. It can be written both ways. Okay, organ identity genes include AP1, AP2, AP3, pistillata, and agamus. Okay, so agamus when it is alone, it will form carpel. Pistillata, AP3, and agamus all together will form stamen. Apetala 2 and AP3 will form petal, and apetala alone will form sepal. Clear? So gibberellic acid pathway activates agamus like 20. Okay, so gibberellin helps in promotion of flowering. This we can see from here. Now vernalization inhibits FLC gene. Now FLC gene, this have the inhibitory role in flowering. So vernalization inactivates FLC gene. Autonomous pathway, GA pathway, photoperiodic pathway, all are indirectly inhibiting FLC. FLC when it's inhibited, then agamus like 20 can be activated. Clear? Now look at this pathway. This the one I have drawn was a simplified version of this only. So see, sucrose concentration, gibberellic acid, low temperature which is vernalization, leaf number which is activating a pathway known as autonomous pathway. They all, this low leaf number and low temperature, they are inhibiting the flowering locus T, because flowering locus C is an inhibitory gene for flowering. So once this FLC is inhibited, they activate what? They not activate. Once this flowering locus C is inactivated, the agamus like 20 gene is released from deactivation. It is activated. Now this agamus like 20 is activating leafy, and they both are meristem identity genes. Meristem identity genes have the role in the transition from the shoot to floral meristem. Shoot apical meristem to floral meristem. Okay, this leafy gene is activating what? Floral homeotic genes or floral organ identity genes. Okay, which will form floral organs. Now, photoperiodism, cryptochrome, all what they are activating? They are activating the gene constans. This constans is again activating agamus like 20. So this pathway is again very, very important. Remember, the lot of question comes from constans. Constans is a activation, helps in activation of flowering, or helps in promotion of flowering. Now, let's start with the question. Based on ABC model, what would be the structure of flower that had normal expression of gene A and C and expression of gene B in all four holes? Okay. So it's saying that if I draw here, this is A, this is C, and it's saying that B is activated everywhere. Okay. So here this region where the B is activated. Okay. So this region becomes A plus B. So I can say this region is A plus B. This region is also A plus B. This region is B plus C. This region is B plus C. A plus B forms what? A plus B forms petal. A plus B forms petal. B plus C forms stamen. And B plus C forms stamen. So is there any similar answer? Yes. Stamen, semen, petal, petal. Clear? Another question. So answer is written here, but still let's find out. Oh, okay, answer is written, then not going to solve here. You try to find out whether you can solve it or not. Next, based on ABC model during flower development, loss of class A activity results in the formation of only stamen and carpel. Which of the following floral organ identity genes control the class A activity? We already discussed plenty of times that Apetala 1 and Apetala 2 controls class A gene. Coming to the next question, according to the ABC model of floral development in Arabidopsis, as shown below, so this is the wild type 1. Several gene transcription factors AP1, AP2, AP3, gammas, etc. are involved. Which one of the following is correct? Apetala 2 transcript expressed during sepal and petal development. Which one is correct? We need to find out. Gammas AG is considered as class A gene. I told class A gene expresses what? Apetala 1 and Apetala 2. Here I write again separately. Apetala 1 and Apetala 2 are expressed in class A gene. Okay, so this is wrong. Apetala 1 is expressed during carpel development. No, for carpel development is gammas. Wrong. Apetala 3 expressed during sepal development. For sepal development, I told AP1 and AP2. Okay. Again, I write for this also. It is Apetala 2, Apetala 3, and pistillata. And B plus C is Apetala 3, pistillata, and ergamus. Clear? Next. So individual and overlapping expression of homeotic genes in adjacent holes of a flower determine the pattern of floral organ development. In Arabidopsis mutant, floral organs are distributed as follows. So students, all the questions are from the mutation of ABC genes. If you just give concentration while solving this, you can solve all the questions, I'm very sure. Okay, so hole 1 will produce carpel. So it is producing what? Try to solve it on your own. So carpel, stamen, stamen, and carpel. That means there is no formation of petal. Okay. So loss of function mutation in which of the following gene would have caused the above? So stamen is formed. That means Apetala 3, pistillata, and ergamus is expressed. Because stamen, which is B plus C, requires these genes. So they are not mutated. Apetala 3, pistillata, and ergamus are not mutated. Okay, because stamens are expressed. Carpel require only ergamus. But this sepal and petal, sepal require what? Sepal require Apetala 1 and Apetala 2. Clear? So this is mutated. Another question, which one of the following statement about leafy is a regulatory gene in Arabidopsis thaliana is correct? So leafy, since the name says leafy, do not get confused that it is keeping the vegetative identity. No, it helps in floral meristem identity. That is the transition from shoot to floral meristem, shoot meristem to floral meristem. Okay, so leafy is involved in floral meristem identity. We come to the next question. Now solve this. Which one of the floral holes is affected in Apetala 3 and pistillata? So I have solved several times AP3 and pistillata. Again I write for you. A is Apetala 1 and Apetala 2. A forms what? Sepals. I'm writing for the last time, okay? A plus B have the genes what? Apetala 2, Apetala 3 and pistillata. A plus B forms petals. Now learn by heart. I've written several times. Next is, wait. Next will be, write on your own. Next will be B plus C. Wait. Next will be B plus C. B plus C will form what? Like what genes are required? Petala 3, pistillata, and ergamus, which will form stamens. So according to the explanation I have written, find out the correct answer. Okay, so which of the following four holes is affected in Petala 3 pistillata? So this will be petals and stamens. Clear? Now the last question which we are going to solve now. Rest of the questions you will solve by your own, and if any problem you will face, you post the questions in the comment box or you can ask me in the chat box. Okay. The control of flowering is a complex process involving several key regulatory genes. Some statements on flower development are given below. Two major types of genes regulate floral development, which are meristem identity genes and floral organ identity genes. The important genes in Arabidopsis that play key regulatory roles in meristem identity are Petala 1, leafy, suppressor of constans 1. Okay, the genes that determine floral organ identity genes were discovered as floral homeotic mutants. Okay, so we have to find out which of the following combination is correct. So first let me read. Most plant homeotic genes belong to a class of related sequence known as fad box genes. Did we find fad box? It was mad box. So D is wrong. So how you will solve the question? D is wrong, D is wrong, D is wrong. Only one remains, so ABC is correct. Such a simple one. Clear? So here we complete. I hope this class will be very helpful for each and everyone. Please follow what I have said and write the notes and make the notes of the mutants and the wild type genes in your copy. It will help you a lot to solve all the questions. Whatever questions is there, like be it how much questions are there in your book, go and solve all the questions related to ABC flower development. At least you will score 4 marks for sure. Okay, because every time one question comes from this topic. One, it can be one from part B and one from part C, or especially in part C you will find every time one question is there which is based on the same concept. Okay, so thank you and bye bye. All the best for your exams.